All right, ladies and gentlemen, we are getting ready for this next assessment. So this is how I'm going to grade it, looking at mastery, proficiency, and approaching. And ultimately, we need to describe the general structure and function of enzymes. But in order to do that, we have these three things: description of lock and key model, plan and conduct an investigation on reaction rates, and describe these things using graphical data. So to pass, we want to look at part one first. And when we look at the test, this is part one. So it's draw and label. It tells you the things you have to do, but then I just leave it all blank. And you need to give me a lock and key model. So this is showing a synthesis reaction, but it could be a decomposition. We've done different versions of that. You need to label what is what and tell me what they are, and overall tell me why is it called lock and key. So you have this. I gave it to you as in a private message after formative assessment number one. We also have various versions of this drawn in our notes as well. So that's our lock and key model. So if we come back to, if you're ready for the summative assessment, we go to part two. And that gets into when given a scientific question involving enzymes and reaction rates, can you state independent, dependent constants and a hypothesis? And then can you write a procedure? So again, if we look quickly at your test, I give you a little background information, I give you a question, and then it's set up the same way we've done it in class. However, there is a modification here. And I'm going to spend a whole nother video here talking about formative assessment number two and walk you through that instead of just giving you an answer key. And I'm changing the format down here a little bit. So instead of having separate boxes, it's just one big area with a checklist of the things I'm looking for in order to pass. So as far as that goes, again, I'm going to make another video talking all about this and hopefully help you guys with the procedure part, since that seems to be a bit of a sticking point. So before I move on to the third part of the test, what I want to do is quickly hit this part. We had this assessment where we were supposed to go through these, and we've done this as a do now in the beginning of class. But again, you have a question. Does the independent variable affect the dependent? And here I've color-coded these things. So what comes between the does and the affect is the independent variable, and that's what we are going to modify or manipulate or change in our experimental setup. The sweating is the dependent variable. We're going to measure this as the result, and it's going to be the data involved with our experiment, that after we do the experiment, we're collecting this data. Constants, and some of you guys are getting to the point where you're trying to look for all of this in the question. The constants or the controlled variables are things that you need to use your imagination and start thinking about the actual, how this experiment would look in order to come up with constants. And when I talk about constants, I'm thinking of everything that could possibly affect our data that we would want to be the same. We want to only isolate the modification of temperature. And all of these other things should be the same for each person we test this on. They should be the same age, they should have the same activity level, the same body mass index, hydration level coming in, the amount of clothing on should all be the same. Now, as we come up with our hypothesis, there's many different formats. The biggest thing I want to see is that you're giving me the trend. So as you increase the independent variable, or as you change the independent variable, tell me what's going to happen to the dependent. So here is my prediction. As the temperature increases, the sweating will increase. It's a prediction about the trend in the results, and that's all you need. So tie that back into our own enzyme stuff right here. We're looking at pH and the time it takes for milk to turn into cheese. And again, this is going to get into our cheese experiment. So the independent variable is pH. It's what we would manipulate or modify in the setup. When we have multiple batches of cheese, we're going to change the pH in each one. However, the temperature of the milk, the type of milk, the amount of enzyme, how you stir it, all of those things would be the same for every batch of milk. But each batch of milk would have a different pH, and the different part is the independent variable. What our results are going to be our dependent, the time. Again, make a prediction as the pH increases, the time to make the cheese will increase. I use time here, but as far as your assessment goes and your summative and all that kind of stuff, we're always talking about rate of reaction, which gets into this. And rate is just how fast something goes, as far how fast how something changes over time. So, does the temperature of the enzyme affect how fast the reaction happens? In this experiment or this question, we need to first isolate all of these things before we come up with our procedure. So again here, if we're looking at the independent variable, it should be temperature. Looking at the dependent variable, it should be the speed of the reaction or the reaction rate. The constants, what else could affect reaction rate besides temperature? It would be pH, the amount of the enzyme, the type of the enzyme, the amount of the substrate, what we're starting with, the size and the type of container that the reaction will take place in should all be the same, and the same is constants. Those things are not necessarily found in the question. You have to use your imagination to come up with them. And now with our hypothesis, give me the trend that you would expect as the temperature, because that's our independent. As we increase the temperature, the rate of reaction should also increase. And then there's going to be a point at which we break down the proteins and all reactions stop at that point because the enzyme can't help. Okay, so that's the second part. And again, going over the second formative, I'll give a whole nother video for that. But our last part of the test is to sketch your own graphs for conclusions. And there's going to be 3. So if we're looking at our final test here, I'll give you 3 conclusions. And for each of those, you have to sketch a graph, and your graph has to include certain things. And I'll give you the list of things you need to have there. So if we're looking at this, again, we did this as a do now, but I want you to be able to see it again. So no chemical reaction is possible below a pH of 4. As the pH increases, the rate of reaction also increases until it hits the optimal pH at 8. Above 11, chemical reactions are not possible. That's what I'm going to give you, and you have to give me everything else. And you can see I'll give you the checkboxes of what you need to make sure you have because I'm just such a nice guy. But if you're asked to make a graph, what I need to see is that you have an X and a Y axis. And then you need to put the proper labels on those axes. You need to have a title that includes the independent and dependent variables. If you have a number line, it has to be put on appropriately. Now, we don't have numbers, we don't have the actual data for the rate of reaction, but we just have things like it happens, it doesn't happen, optimal, but it doesn't give us specific numbers for like substrates per second. So we're just showing the general trend in the line that we put on there. It's going to be line graphs. But based off of the summary, we would start at 4 because below 4, nothing is possible. The optimal is 8, so then the line would peak at 8. And then it says above 11, they're not possible. So then the line would also dip at 11. And again, I give you different scenarios about possibly plateauing or denaturing and stopping chemical reactions and gives trends and all that kind of stuff. So again, that's the third part. You don't have to do this very well, and you can still pass the standard. The things that are absolutely necessary to pass the standard, though, are this and all this kind of stuff, where I give you the question, you give me independent, dependent, constants, hypothesis, and write out a procedure. So hopefully there are no questions as far as what the test is going to look like and what you have to do to pass. However, if you need extra help, make sure you come and talk to me.